the first talk after the MOB meeting in Australia. So Elena did her PhD at the University of Southampton in DTP at the National Oceanographic Centre. She graduated in 2018 and then moved to a position as a research fellow at National University of Singapore in the group of Danwei Huang, where she stayed until 2021, where she decided to move back to Europe as a postdoc in Michael Kult's lab at the University of Copenhagen. So Elena is doing great work on the biology of corals, and today she's going to tell us about microscale light management by coral pigments. So please, Elena, take it away.
Yes, thank you so much, and thank you for inviting me to give this talk. So for those who I didn't have the pleasure to meet at the conference, I'm a marine biologist and ecophysiologist, and I have spent about the last 10 years studying coral reefs, and in particular, the processes and the interactions that occur at the very small scale inside corals. Is this going to work? Yes. Yeah, so sometimes when we research things at the very small scale, I think we forget to mention why our research is important on the very large scale. So I just borrowed this slide with some estimates that are now actually quite old about the value and the benefits provided by coral reefs. So they support a huge proportion of marine biodiversity, including about a quarter of all known fish species. They're a major source of income and of nutrition as well, particularly protein, for developing countries. They also physically protect coastlines from waves and storms, and they're a very prolific source of new natural products, including many drugs, but also including some molecules that are very important in biotechnology, including the pigments that I'm going to talk about today. And so coral reefs are built by corals, which are animals related to jellyfish. And these are mostly sessile polyps. So they're attached to the substrate and they cannot move around. And they form the reef structure by secreting a hard calcium carbonate skeleton. So corals can reproduce sexually, actually, and baby corals exist as a free-swimming larvae. And then when these larvae find a suitable substrate to settle on, they undergo metamorphosis and they reach the polyp life stage, which is the typical coral life stage. And so for most species, this single settled polyp then begins to reproduce asexually. So it splits into two, and so on and so forth until a colony is formed. So most reef-building corals exist as colonies of individual polyps, which are genetically identical. Now, inside the tissue of these corals live a group of symbiotic algae. And these are called Symbiodiniaceae, they're dinoflagellates. And they, being algae, they fix carbon via photosynthesis, and then they translocate a lot of this carbon to the coral host, to the animal cells. And this is in the form of glucose. And this glucose provides the majority of the nutritional requirements of the coral host. So it's basically the main source of food for the coral animal. And it's also really the main energy source that supports all life on coral reefs. You can see here, that's my laser pointer, you can see the algal cells imaged as chlorophyll fluorescence inside the tissue of a coral polyp here in light sheet microscopy. And so if we take a look at the microscale context that these coral symbionts experience, this is a schematic of coral tissue. It's a very simple animal, and it's composed of an ectodermal tissue layer, which is the most external one facing the seawater. Then we have the symbiont cells residing inside the coral gastrodermal tissue. And this gastrodermal tissue lines the gastrovascular cavity. through which nutrients and different products can be exchanged between polyps. And on the other side of the gastrovascular cavity, we have another layer of gastrodermis containing the symbionts. And then we have the arboreal ectoderm. And this is a specialized type of ectoderm which secretes and attaches to the coral skeleton. So basically, coral symbionts live inside coral cells, and they're sandwiched between a layer of ectodermal tissue, so host tissue on the outside, and a layer of calcium carbonate skeleton underneath. And the reason I'm mentioning this is that this whole structure is key in determining the light microenvironment that these symbiont cells experience, and therefore the amount and the type of light that is available for photosynthesis. And light availability on coral reefs exhibits a huge range. We go from this kind of environment, which is extremely well lit. This is a reef crest, or the reef flat, like we saw on Heron Island, that experiences supersaturating light intensities of thousands of micromoles of photons per meter square per second. And we go all the way down to mesophotic coral reefs, and these are, depending on the region of the world, this can be beyond 100 meters depth. And in this situation, we have light as a limiting factor for photosynthesis and for growth of corals. And as well as this gradient in light quantity, we also have a change from a change in quality of light from a broad spectrum in shallow water, which includes quite a lot of UV and also infrared, down to a narrowing of the spectrum towards mesophotic depths, where we have mostly just blue light really dominating, and there's still some UV, but UV penetration is greatly reduced. And so corals, they're sessile, cannot move, and so they have evolved a range of mechanisms to help them support and fine-tune photosynthesis at both this high range and this low range of the light distribution. And one example is the plasticity of their skeletal morphology. So here we see pictures of coral colonies from the same species in each line, and the form that they assume in shallow water versus mesophotic. And this difference in skeletal structure translates to a difference in the light gathering ability of the coral. So shallow water morphologies tend to be less good at gathering light. So they kind of reduce how much light reaches the symbiont, whilst these mesophotic forms tend to have as much light gathering ability as possible. And so what a lot of my research has focused on is what role coral pigments have in this context. So how do coral pigments help corals optimize photosynthesis and the internal light environment of corals at both ends of the light spectrum? So both the coral host, the animal, and the symbionts contain pigments. The symbionts, of course, contain photosynthetic pigments such as chlorophyll and peridinin. These are used to do photosynthesis, and they give corals kind of brownish, greenish coloration. But the host tissue can also be very heavily pigmented, actually. And in particular, it contains these very bright pigments known as GFP-like proteins. And these pigments are what makes coral reefs so colorful. So it gives them these vibrant yellows and greens and pinks and purple. I believe many of you might have seen these very colorful coral colonies on Heron Island following a bleaching event. And I'll talk more about that soon. So these host pigments are homologous to the green fluorescent protein. which is a protein that some of you might be familiar with. It was discovered in the 60s in the jellyfish Aquaria Victoria. It is capable of absorbing light of a certain wavelength, let's say blue, and re-emit it as light of a slightly longer wavelength. So it emits fluorescence. And the green fluorescent protein became very famous because it can be easily expressed in recombinant organisms. And so it became very widely used in live cell imaging and in biotechnology. And it earned its discoverers the 2008 Nobel Prize in chemistry. So famous protein. Yeah, so the reason it became so famous is that it's very easy to express in recombinant organisms such as E. coli that you see here, or even, why did my mouse not work? Sorry. Even in more complex organisms, including mammals. And actually, corals played a very important role in this biotechnology revolution, thanks to some of their fluorescent proteins, because we went from having just the green color that was provided by jellyfish fluorescent proteins to a whole array of new colors. So red fluorescent proteins are very common in corals. And thanks to these proteins, we're now able to do, for example, dual labeling inside cells. So corals, yes, have helped biotechnology a lot. So coral GFP-like proteins have a structure that is very similar to the jellyfish GFP. Their main structure is a barrel of beta sheets, and most coral fluorescent proteins have 4 identical subunits of this kind of original jellyfish structure, let's say. The barrel surrounds the chromophore, which is the light-emitting part of the protein. And yes, and most GFPs that are used in biotechnology are made monomeric, so they have a single subunit. But coral GFPs, as I was saying, tend to have 4 subunits, so they're tetramers. There is another important difference between the coral GFP, between the jellyfish GFP and coral GFPs, is that the jellyfish GFP exists predominantly with a chromophore form that absorbs UV light. So it is a protonated chromophore. And the deprotonated form of the chromophore has this blue absorption peak here. But the jellyfish GFP mostly absorbs UV light. Coral GFPs, on the other hand, have chromophores that exist predominantly in the anionic form. So they tend to absorb visible light. So this is something that in the coral literature is very often misunderstood, because people talk about how coral GFPs absorb UV light, and actually their UV absorption is relatively limited. So yes, they most absorb and emit visible light, but their spectra are very diverse. You can see here proteins that emit cyan, green, orange, red, and you can also see some of these proteins absorb light very strongly, but they don't emit any fluorescence. And we call these chromoproteins. They have pretty much the same identical structure as fluorescent proteins, but they emit very little or no fluorescence. So people tend to use colors to classify coral fluorescent proteins and chromoproteins. But I would argue that being a biologist, color is actually a terrible way of classifying things. It's like when someone says, which car are you trying to describe? And you say the red one, that's clearly an indication that we don't know very much about cars. And it's the same thing with coral fluorescent proteins. I think there are much better ways of trying to classify them. So for example, we can think about classifying them in terms of how they're regulated. So in terms of their regulation, we can divide coral fluorescent proteins or GFP-like proteins into two groups. The first groups we call light-induced proteins. And these proteins are regulated at the transcriptional level by blue light intensity. So if you keep a coral that can produce this protein under high light and high blue light in particular, it will accumulate lots of protein. This is regulated at the mRNA level. Whilst if you keep the same coral under low light, it will have low concentration of the protein. But there are also proteins that might have a very similar spectrum, such as this protein here has the same spectrum as this one. But their production is independent of light levels. So a coral grown on the high light and a coral on the low light will produce the same amount of protein. And there's also a second regulation mechanism that is actually not very well studied. Yeah, we don't know so much about it, but some of these proteins appear to be downregulated in response to heat. So these two pictures, in the second picture, there is actually a coral that has been exposed to heat stress, but it looks completely black in the fluorescence image because the fluorescence is almost completely gone as a result of the heat stress. And another important way of classifying these proteins is according to their distribution. So many of them are found in the ectoderm, as you can see here in these sections. This is the coral ectoderm, and you can see it's pretty much all full of this red or orange fluorescent protein. But sometimes we also find coral fluorescent proteins in the coral gastrodermis. For example, here we can see this long wave green FP that is distributed around the zooxanthellae, around the algal symbionts, and same in this image. And so again, this is likely important in terms of function and light management by these proteins. And a final difference in distribution is whether these proteins are diffuse or granular. And so in this image, similar to here, the protein is just distributed in the cytoplasm quite diffusely. And in other corals, you can see here, it's tightly arranged in granules. And these granules are strongly scattering. And so proteins that are distributed in these two different ways might again have different implications for light management and for function. So the function of these proteins in corals has actually been historically a little debated, but I think it's because they are so diverse in their optical properties, in the distribution, in the regulation, and this is a likely indication that they don't all have a single function. So these are the functions that have been proposed. Today, I'm only going to talk about photoprotection and light enhancement, as these are the ones that deal with light management. But I just wanted to put this out there to make it clear that we do have evidence for some other functions as well. So there's probably more to the story. So my work has focused mainly on two groups of proteins. One are light-induced proteins that are found in shallow water corals. So these are the ones that increase production in response to light. And the second one is a group of light-independent proteins or constitutive proteins that are found in low-light adapted corals. For example, mesophotic corals. Oops. And I'm going to start from the second one because that's apparently the order of my slides. So one type of protein that is very common in depth generalist corals, including mesophotic corals, is this type of orange protein found here in this lobophilia. And this is actually the same image that I was showing you to show the ectodermal distribution. And this is quite an interesting protein because when it's first synthesized by the coral, it appears green. So it has an anionic form of the chromophore that absorbs blue light and emits green light. However, the neutral form of the chromophore, which, if you remember, absorbs UV, when absorbing a UV photon, actually undergoes a complete transformation of the chromophore. So there is a physical break in the peptide backbone, which is irreversible, and it causes the pi conjugation to extend to this histidine residue here. And by extending the pi conjugation, we change the excitation and emission wavelength of the protein. So when this protein is first synthesized, it's green, but then upon absorption of a UV photon, it becomes a red fluorescent protein. And we call these photoconvertible fluorescent proteins, and they're actually quite widely used in biotechnology. And I think this is quite interesting if you think about these proteins existing in a mesophotic environment, because we know UV is attenuated by the water column. And so the photoconversion rate of these proteins, in theory, should decrease exponentially as you get deeper. And these are measurements done in the lab on clone proteins. But what I think is interesting here is that photoconversion is actually very efficient in these proteins. So even under very low UV irradiance, you can still get significant photoconversion after a relatively short time. I think this graph here is about an hour. So photoconversion rate changes with depth, but we can, in theory, still get significant photoconversion at depth. So we wanted to see what kind of phenotype you would get from corals grown under different amounts of UV. And so here we have a coral that, this is before and after, corals that have been exposed to an amount of UV that is equivalent to a shallow water reef. No UV, which is something that, according to our calculation, is beyond the depth limit of this coral species. And also an intermediate amount of UV, similar to what you would measure on a mesophotic reef. So under high UV, the coral is mostly red. You can see the spectrum here. Under no UV, the coral is almost completely green because you get accumulation of the unconverted protein. So you have pretty much no red anymore. But under mesophotic conditions, the protein pool is maintained in this partially photoconverted state. You can see here the kind of almost equivalent peaks in the green and the red emission and the color that looks kind of yellow. And this partially photoconverted state is dictated by the balance between the amount of UV photons present in the environment, which makes the protein convert, and the turnover rate of the protein, which basically gets rid of the old red protein and replaces it with new green protein. And so I want to look a little deeper into what it means when I say that the protein pool is partially converted. So remember I mentioned that these proteins are tetramers, so they have 4 identical subunits, each with its own green chromophore to start with. And it takes 1 UV photon and 1 photoconversion event to get turn each chromophore to the red emitting form. So you need a second photon to convert the next one, excuse me, and so on. Now, when we have this kind of configuration, the protein can actually absorb a photon through the green chromophore, and then it can transfer this excitation energy directly to the red chromophore via fret. So this is a non-radiative form of transfer. And then the red subunit will emit a red photon. And this mechanism is actually 100% efficient. So as soon as you have a single red subunit inside a tetramer, the whole tetramer will only emit red fluorescence. Green fluorescence emission only occurs if the tetramer is fully green. So overall, basically, this is the only green emitting tetramer, and all the others just emit red light. And this turns out is an incredibly efficient mechanism to convert this cyan green light into orange red light. So it's quite a long range wavelength transformation that is incredibly efficient. If you look at how much orange red fluorescence is emitted per microgram of protein, you see that this partially photoconverted protein pool has the highest efficiency. And just for comparison, this is a protein that has the same spectral properties. So it can also convert from green, it can also, absorb in the green and emit in the red, but without having this dual fluorophore mechanism. And its wavelength conversion efficiency, it's much lower compared to the partially converted photoconvertible protein. So what does this mean for corals? Why is it interesting? And it was previously proposed that the orange fluorescence emitted by these proteins could enhance photosynthesis in deeper areas of coral tissue, because orange light is poorly absorbed by coral symbionts, and this allows deeper penetration inside the tissue, which is equivalent to what you see with green light stimulating chloroplasts deeper in a leaf, or in a bioreactor, giving green light instead of blue light stimulates photosynthesis in deeper algae layers. However, all the evidence we had of this was from this was done in anemone tissue or estimated using calculations. And we didn't actually have any confirmation that this is actually what happens in corals. So we wanted to measure how do these proteins and the photoconversion process affect the light environment inside corals in vivo. And so we took a colomorph of this coral, Montastra cavernosa, the same one I showed you before, that contains the photoconvertible protein. And we used a scalar irradiance microsensor to characterize the light environment inside the tissue underneath the host pigment layer. And for those who were at the workshop on Heron Island, this is the same setup that Swati was using to measure light on the surface of corals. But in this case, we used a needle to pierce a hole in the coral tissue, and then we could collect light profiles inside these corals, which are a little thicker tissue. And this was work done in collaboration with Daniel, who was in Michael's lab at the time. So first of all, we performed these measurements using white light. You can see here the scalar irradiance spectrum shows the absorption and emission signatures of both the green and the red form of the protein. And if we look at the attenuation coefficient through the tissue, we see the overall overall orange red light is less attenuated by corals, whilst green and blue are more attenuated. But there's really not much difference between the coral that has the more converted protein and the coral that has the more unconverted protein. But if you think about what this means for corals and mesophotic reefs, we perform these measurements with white light, but actually light is not white on a mesophotic reef. We know that this part of the spectrum actually disappears as we move deeper. And so we took out our broad white measuring light and we replaced it with a blue light, which simulates a little better the kind of spectrum that these corals experience in the field. And when you only provide corals with blue light, you can see how fluorescence is distributed in the tissue. So this is, there's no incident in this area. So this is just emitted fluorescence. And you can see that in the unconverted coral, you have green fluorescence penetration here. And as the coral becomes progressively more converted, you see the slight peak in the red forming. And as we get to the most converted coral, you see the red light actually penetrates much better, or orange red light penetrates much better in the coral tissue. And it can reach very well the full depth of the coral, even though the site of the emission is in the ectoderm, so at the top here. So we try to quantify this in terms of photons. So basically, for each blue photon that reaches the coral surface, how many green or orange photons arrive at depth inside the coral tissue. And again, just repeating, this is only from fluorescence emission. There is no light in this range in the incident that we used for these measurements. So in the most converted coral that we were able to look at, for 100 incident photons of blue light, you can measure about 5 orange photon at the tissue surface and about 1 orange photon that travels all the way to the deepest point that we measured. And so this may look like a very small amount of photons, and it is. So this is possibly why we don't see a difference between the unconverted and the converted coral when we measure on the white light, because there are so many orange photons in white light that this completely falls within the noise. But when we move into an environment where there is no orange light, actually the ability to produce even this small amount of orange light might become a small fitness advantage for the coral. And so we try to put this into the environmental context of a mesophotic reef. And so we used some published data of irradiance collected in the field in the Red Sea. And for each depth, we did a calculation, a rough calculation, of how much orange light a coral would get from downwelling sunlight versus how much it would get from the fluorescence that it produces. And you can see that as you get deeper, the relative contribution changes, and below 80 meters, coral fluorescence becomes pretty much the only source of orange light that the symbionts will experience. And again, I just want to make it quite clear that even though this is the main or only source of orange light inside the tissue, this is still a very small amount of light that is produced by the coral. And so the effect that it could have on photosynthesis and any potential fitness advantage for the coral is still likely very small. And it may even be counteracted by the metabolic investment required to maintain these proteins. And this makes sense because if this was giving mesophotic corals a huge fitness advantage, then all mesophotic corals would be red. And we know that that is not the case. But we do actually have evidence from a previous study that show that at least for some species, if you take colomorphs that contain the orange protein and put them under low blue light for a long time next to corals of the same species that don't contain the orange pigment, the pigmented corals actually survive and grow much better. So what we see here could be perhaps the result of those few orange photons and that small fitness advantage that allows corals to survive at depth. Of course, this opens up a lot of challenges related to trying to actually measure this potential small effect of orange fluorescence on photosynthesis, particularly inside a living organism or one day in situ. But we have been working on this, so hopefully we will have more results on this to present soon. So now I'm going to move on to the other end of the spectrum, which is looking at corals that live in these highlight shallow water environments, and try to look at the function of some of these light-induced proteins. So in shallow water environments, the challenge is quite the opposite. Corals are exposed to excessive light levels, which can actually damage the photosynthetic machinery of the symbionts. And if we combine these high light levels with the recurring episodes of heat stress that are caused by climate change, we get bleaching events. So the symbiosis breaks down and the coral tissue loses most or all of its symbionts. So the loss of symbiont pigments is what makes corals appear white in the image, and is what gives the name bleaching. However, sometimes this phenomenon actually happens, and we saw some examples of this on the heron reefs over the past month, which was interesting. So these are still bleached corals, but they appear very colorful instead of white. And this is quite clearly caused by host pigments. But at the time, we didn't really know what the mechanism behind this was. Or why doesn't this happen every time there is a bleaching event? And is there a function, is there a reason why these corals become so colorful during bleaching? And one hypothesis was that the host pigments just become more visible when the symbionts are lost, because the white skeleton and the tissue are very reflective. And so the light emitted or scattered by the pigments is enhanced and it's made more visible. So basically, it's just an optical effect given by the loss of the symbionts. However, when we looked at it more closely in bleaching experiments, here you can see a coral before and after heat stress, and here's a measure of bleaching shown by a drop in the intensity of chlorophyll fluorescence over time. In the dark bars is the treatment. We saw that actually, if we look at green fluorescence emission, which is again here in the dark bars, if this was simply due to an optical effect, we should see an increase in green fluorescence that matches exactly or mirrors the decrease in chlorophyll fluorescence. And instead, we see a slow, gradual ramping up that happens after bleaching has occurred. So this to me, this is actually a graph from my master's thesis many years ago. And it was very exciting to see that this actually looked like a form of biological regulation instead, and not just an optical effect. And so I went on to repeat the experiment quite a few times and quantified the protein by extracting it from the tissue. And indeed, you can see that whilst at the beginning you see an increase in green fluorescence that is not matched by an increase in protein, at the second time point, we have a much higher protein concentration. So indeed, there was an increase in protein production in bleached corals. And the biggest increase in fluorescence actually happened during the recovery phase. So when the coral was still bleached, but the heat stress had been removed. And we also looked at other form of stressors that induce bleaching. This is nutrient stress, looking both at fluorescent protein and a chromoprotein. And I think this picture is quite striking because these two corals are clones. And so I find it crazy that they can just look so different. Anyways, we kept seeing the same response. And so another piece of the puzzle came from this experiment, which was performed by Rachel. And she bleached some corals, which we know can produce a green fluorescent protein or a pink chromoprotein in this case. And then she kept some of these corals under green light for 3 weeks, which did not cause the colorful bleaching phenotype. And then she kept some corals under a blue light, which instead did cause the colorful bleaching phenotype. And so what we think is happening is that if you recall that some of these pigments are regulated at the transcriptional level by blue light intensity. Now, inside a healthy coral, we have low blue light fluxes because blue light is absorbed by the symbiont cells. Now, once the coral is bleached, these blue light fluxes increase massively, and light is reflected by the skeleton and amplified throughout the tissue. And so we get this transcriptional cue that causes the upregulation of the fluorescent proteins or the chromoproteins, and we end up with this colorful bleached phenotype. And the next question is, why do they do it? Does it have an evolutionary advantage? Because making so much of one specific protein at a time when the coral is pretty much starving and it has no food, it could be very detrimental if it doesn't have some kind of benefit. And so, again, we perform the same microsensor measurements, looking this time at a brown coral without the pink pigment and a bleached coral that had high concentrations of the pink pigment. And we saw that the corals that don't have the colorful phenotype, the tissue is generally light enhancing in the top layer. And then light is attenuated as we move down, particularly in the absorption range of photosynthetic pigments. But if we look at the colorful bleached coral, we see that within the absorption spectrum of this pink chromoprotein, actually there is massive shading throughout the tissue, all the way down to the skeleton. And so clearly this reduces light stress inside the tissue, which likely reduces any further photo damage for symbiont cells that might be left, but it might also make it easier for new symbionts to come and recolonize the tissue. And we did see that for corals that have higher concentration of the pink chromoprotein, which is the pink bars here, when allowed to recover under white light, they regain the number of symbiont cells and the photosynthetic efficiency of the symbiont cells faster than corals that don't have as much of the pigment. And so we think that what's happening is that once this pigment layer becomes established, it then reduces light fluxes inside the tissue, and this reduces light stress, and it allows for new symbiont cells to come and colonize the tissue. And then once the symbiont cells start to move into the tissue, then blue light fluxes are reduced again, the transcriptional signal goes down, and then the coral regains its original state. So it's an optical feedback loop. And then the last question that I'm left with is, why does this not happen every time there's a bleaching event? And why don't all corals do it? And so we tried to compile some anecdotal evidence of colorful bleaching events from around the world. And we tried to get some climatology data surrounding them. And we saw that these reports of colorful bleaching usually came after a short episode of heat stress, after the temperature. So this is when the pictures were taken. So after the temperature had dropped back down below the bleaching threshold. Or in other cases, we saw reports of colorful bleaching during milder heat stress events, or at the beginning or in the milder phase of more severe heat stress events. And this is in agreement with what I mentioned before about some of these proteins being downregulated in response to heat stress. So there is probably an interplay between this heat-induced downregulation of the protein and the light-induced upregulation of the protein that results from bleaching. And I think this is an important message in terms of climate change, because it shows that it's not just about whether or not we have a bleaching event on a given year that will determine coral survival, but the intensity of the stress can also affect the potential recovery mechanisms down the line. And finally, what about corals that never do it? We know that you can find on the reef colonies of the same species side by side, and one has the pigment and one doesn't. And so we think that there is some kind of trade-off of pigment production, perhaps the fact that it requires some metabolic investment to maintain a high level of these pigments. So maybe at times when conditions are not so stressful, it is advantageous to not produce the pigment in terms of growth. And we also thought that there could be a trade-off in terms of heat production by these pigments, because they absorb light so strongly. And this is what Swati has been testing on heron over the past month. So you can ask her more about it, and definitely stay tuned for her results. And so, yeah, I guess the message that I want to conclude on is that, like I mentioned, for photoconvertible proteins, whatever fitness advantage is given by these pigments or any other, polymorphic mechanisms in corals, the advantage is likely to be small, and there are likely some trade-offs. So if we're trying to assign function to these mechanisms by looking for a huge fitness advantage that outcompetes all other phenotypes under all conditions, I don't think we're going to find that. I think evolution would have found that a long time ago. But I think that what these proteins remind us is that the diversity of mechanisms and phenotypes and genotypes that have allowed corals to survive in different environments and changing conditions is huge. And even a small advantage like this might be what helps corals get through the next century. So we need to keep looking. And yeah, that was the end of my talk. Thanks to all my co-authors, and thank you again for inviting me to speak. And please, if you have any questions, I'll try and answer.
Thank you, Elena, for a really fascinating talk. Are there any questions? Please raise your hand or just unmute yourself. Jonas?
Hey, thanks, Elena, for the very engaging talk and a very clear one. I have a question about, I know nothing about corals, and so are some of these photoinducible proteins, is this change in morphology reversible?
In the change in protein structure? Yes, it's
Yes.
reversible. This particular one is. There are other types of coral fluorescent proteins that are photoswitchable or kindling. So some of them can become fluorescent and then become non-fluorescent. Some can reversibly change color, but this particular one is a break in the peptide backbone, and it is not reversible.
Oh, it's not. Okay.
Not reversible.
Not reversible. So, when the entire coral changes color, that's also globally not reversible, or the corals can still degrade the proteins and just produce new ones?
Yeah. So the protein is, I think the half-life of the protein is about 14 days.
Okay.
So as the protein turns over, the new protein that is made is green. So depending on how much UV you have in the light field, you end up with these like different degrees of photoconverted corals. Yeah, so it's a balance between the turnover rate of the protein and the photoconversion rate, which changes with depth. And also the photoconversion rate is different for different proteins. So you have some that are very, very efficient. And even on the very low UV, you will find the corals looking very red. And other species have less efficient protein. So they get more of this mix, green and red color.
Last question from me. On a previous slide, you say that corals, same species, can live side by side but have different colors. But there is still some length scale that somehow determines that a given group of polyps have the same color. Like, how is that coordinated spatially?
Yeah, sorry, great question. So these are not clones. So in this image, all of these pink ones are clones, and then all of these brown ones are clones with each other. So you have two different colonies that are not clones, but then within the colony, all of the polyps are clones. So they're two different genotypes, but they are the same species.
Awesome. Thank you.
Thanks.
Any other questions? I have a question actually. Regarding the case in which you have the orange light production at depth, where that's the, if I understood well, is the only source of orange light at that depth, right?
Yeah, for the coral.
Do you think that can be a sort of like a clue for larvae? Do you know whether perhaps they can see light?
Yeah, so I know that larvae are phototactic, but I think not towards red, if I'm not wrong. But when I mentioned the potential different functions of fluorescent proteins, one of them was signaling. And we do actually know that not larvae, but zooplankton prey are phototactic towards orange light. And so sometimes corals will have a ring of the protein around the mouth, and that attracts plankton prey to swim into them. So yeah, that's another one of the main proposed functions of fluorescent proteins, especially orange ones. And there's another one, this was shown for green fluorescence rather than orange, but actually symbionts appear to be phototactic towards green fluorescence. So this could also be involved in the recolonization process, that having this strongly expressed GFP layer might kind of convince the algae to come back into the tissue.
Really fascinating. Nicola?
Yes, hi. I also have a question, but you just mentioned it, Elena. Thanks for this great talk. I was also curious about this phototaxis, and in fact, I wanted to bring up this the same example you mentioned, that symbionts are attracted to green fluorescence. I'm also interested in the other citation or the other work you said about plankton prey being attracted by light. But regarding symbionts, do you think the chromoproteins, or for instance, pink Poecilopora that you mentioned, would it also attract symbionts? Are they maybe attracted to other wavelengths and not only to the green fluorescence?
So I don't think this has been tested. I guess, I mean, it is possible that they're attracted to just bright colors in general. I don't know what kind of light receptors they have. Intuitively, for me, fluorescence is easier to pick up because it's a bit of a different signal, and just the broader reflectance signal, like from a pink protein, might be less specific. But I mean, I don't see why not. I don't think it has been tested before whether pink does attract. But there's also, I mean, it could just be bright colors in general, right? Because they're not all pink. There's lots of purple, blue, red, all sorts of color chromoproteins, and most of them are upregulated like this during a bleaching event. So yeah, maybe just bright colors.
Okay, thanks. And what would be the reference you said for the phytoplankton attraction?
It's Bensby et al. 21.
I'm sure you can also email Elena if you want to
feel free.
set up the references. Is there anybody else that would like to ask a question to Elena? Okay, if not, then let's thank Elena again. And it was a pleasure to host you here. And next month, there is going to be the last talk before the pause in August. It's going to be given by Corina Broussard from the University of Amsterdam. So thank you again, and see you next time.
Thanks for having me again.